Over 60 different species have faced extirpation or population reduction at the hands of the invasive genus Rattus. Wild populations interfere with ecosystem functionality, trophic level interactions, and general stability. Their presence and overabundance has since been linked to the spread of diseases and multiple different ecosystem collapses. Here in Santa Cruz, we have two primary species of Rattus, the first being Rattus rattus, also known as the black rat, ship rat, or roof rat. The second of the two species is Rattus norvegicus, or the brown rat, Norway rat, or sewer rat. Both species originated from Asia, but they are now found on every continent excluding polar regions. Known to be extremely aggressive, these rats will attack and eat nearly anything that is comparable to their body size. This includes, but is not limited to, eggs, bird or reptile hatchlings, as pictured, lizards, and crabs. Their ability to climb and swim, their generalist diet, and their fear of novel objects makes them incredible invaders. We have documented evidence of both of these species of rat on the UCSC campus. However, they have only been spotted near dining halls or other sources of human food. Very little is known about how the rat's spatial distribution extends beyond human habitation. We tested the hypothesis that Rattus spatial distribution is only loosely constrained by human presence, and that the presence of Rattus is positively associated with forested land cover. In order to test this, we created 7 transects. Each transect consisted of 3 different points, all starting at the location of a dumpster, dining hall, or other source of human food. Male and female rats have an average dispersal rate of 43 and 52 meters respectively, so we placed sites far enough apart so that we wouldn't detect the same individual twice. Keeping these dispersal rates in mind, we then placed the next site, Site 2, back 75 meters in the direction of wild habitat. The third and final site would be located another 75 meters back, for a total of 150 meters away from the initial source of garbage. The sites were located in multiple different types of habitats, including a mixed redwood forest, pure redwood forest, mixed oak forest, and shrubland. Pictured is an example of a pure redwood forest habitat. We placed a camera trap and sunflower seed bait at each site and then collected the cameras after 4 nights. To then test the significance of both habitat and distance from human habitation on rat distribution, we ran an occupancy model in R. We found a significant detection rate in the mixed redwood forest habitat, with a value of -1.742 and a p-value of 0.0096. This lower detection rate could suggest that rats have suppressed activity in a mixed redwood forest as compared to others. Surprisingly, we found no significant occupancy nor detection difference as distance away from human habitation increased. This could suggest evidence of a wild population of rats that already occupy the forest. A wild population of rats would likely cause direct competition between invasive and native small mammal species. However, recent studies suggest that these wild populations of rats will predate on smaller, more docile species of rodents. Pictured is a long-clawed mouse in Argentina that has been repeatedly found partially devoured in rat caches. We on the UCSC campus also have our own species of docile mouse, the California mouse, which plays an integral role in determining forest structure. If the pattern of rat-caused extinctions extends to small mammals, then appropriate conservation action may need to be taken. I'd like to acknowledge all these people for the continued support and help through this project, as it could not have been accomplished without them. I'd also like to thank the Norris Center for funding this project. Thank you so much for listening.